Hello everyone, how are you today? So saving the drowsiest for last. Make sure you all have a good nap before dinner. So I'll do my best to rock you to sleep slowly here. Um, yeah, there's good conversation earlier today, and it's funny, I kind of saw my career flash in front of my eyes all day. I tell you a little bit about myself. I didn't have a bio on the website, but I actually studied economics at University of Guelph in agricultural economics, and then did a sharp turn into fertility. And then I work with micronutrition and plant physiology products, biostimulants. And then I went into fulvic acids, amino acids, bacillus, and all of that. And here I am today with Andermatt, and we do a lot of biological crop protection and some biostimulants as well. So I titled today's presentation Soil Plants and Their Microbes. So it's a bit of a kind of a wrap up for the day. So if you have any questions or anything that doesn't match up with what you heard today, put your hand up, let me know. I don't mind being interrupted. And the goal is to kind of clean up the messaging a little bit. Now with Andermatt, through Hybrix and Bacteria Roots, we focus more on the Bacillus products. So that's where the microbes kind of prime us for that idea. So if any questions so far, you guys ready? Anybody need to run to the bathroom quick? We can all wait. You're good? Okay. Awesome. So microbes, what are they? I love these terms like biologicals and microbes because they encompass a lot of stuff. So it's an easy way out of a small conversation where you can say like, I work with microbes, and then people stop asking questions. So for the purpose of this, for the microbes, we have essentially they're microscopic things that happen in the soil around the plant. So we have bacteria, fungi, that's your mycorrhizae. We have the protozoa, so kind of single cell organisms, and then your kind of nematode type organisms. So they're all microscopic. I have pictures here in case you wonder what they look like under a microscope. There's a rough sketch. So it's all the little things that interact. When we talk about the rhizosphere, when we talk about the biology in the soil, they're all the things that interact with the plant, and they help with nutrient availability. They kind of extend plant physiology beyond the roots of the plant. So they work that synergy with the soil. They can buffer stress and disease. One thing I'm going to talk about with the bacillus is the antifungal properties, how it actually produces antifungal compounds. And then it can build long-term soil fertility. So we talk a lot about feeding the biology, like Glenn talked about this morning, feeding the biology in the soil, making sure there's air in your soil so all the biology can breathe. So that's a very important one. So today we'll focus more on the bacillus. So what are bacillus? Not sure. That's what I'm here for. So we're good. So we're done with the microbes now. I'm not going to talk about anything else, just bacillus. So what are they? Well, that's what they look like. So they look like little pills. And essentially they're gram-positive rod-shaped bacteria. And they're commonly found in soil and plant root zones, so in the rhizosphere. And they're known for promoting plant growth and protecting plants from diseases. So that's kind of what they are. And Andermatt, as a company, we specialize on Bacillus valensis. And it's closely related to Bacillus subtilis, which you might have heard of before. And they're different families. So they're all bacteria. They all vary in shape a little bit. And they each have their own kind of different purposes for the plant. So similar to the amino acids, it's quite complex. There's a lot, but they're all different families, and that's pretty much what we need to remember from this. So some of the key characteristics we're going to go over is so they come in the form of endospores. And that's really cool because it makes them survive harsh conditions. So bacteria can be quite susceptible to UV rays, different temperature changes, different pHs. So the endospores is almost like a little capsule that they live in. They stay in a dormant state. And then when conditions are good and they're going to get fed, they come out and they start multiplying and they make a colony. So, our products are more of a colony forming units. So you can think of it as when the Europeans came to North America, they had the 3 little ships and they were colony forming units, and they came and then they multiplied from there. They produce antimicrobial compounds. We call them metabolites. So essentially the byproduct of the bacteria. And so those metabolites can have positive effects for the plant. And that's where we get a lot of the antifungal compounds. They grow in aerobic environments, so they need air to breathe. So that's where we talked this morning with the PSI. If the PSIs are too high and there's too much compaction in the soil, there's not enough air for the biology to live. And bacteria is one of those microbes that needs air to live. So we need to have aerated soils. And aside from that, they're actually quite highly adaptable to soil ecosystems. So why do they matter to us? Like what is the purpose of the bacillus in agriculture? So as I mentioned, they act as a biocontrol agent. Yep.
Got a question.
Yes.
Lauren had a question.
Oh.
You said the bacillus works as antifungal. How do they differentiate between the good fungal and the bad fungal?
So different bacteria so within the families, there's different strains. And different strains will have different metabolites that they make. And so as a company, it's our job to find the strains that have the metabolites that kill the funguses we don't want. So that's how we pick them. So it's not like it's almost like the amino acid, like certain chains will release certain nutrients, whereas certain strains of metabolites or certain strains of bacteria will create a metabolite that kills a certain fungus. And the second, which I'll talk a little bit later on, but it's the competition idea. So they colonize the root zone so densely that they don't leave room for pathogens to get to the plant. So they act as a biocontrol agent. They reduce the need for chemical pesticides. They enhance nutrient availability for the plants. So we'll talk a little bit about the mode of action for that, and they can be used as biofertilizers. So that's why we care about them. So we talked this morning a lot about the carbon cycle, and I added this slide. I ran up at lunch and added this in because it really resonated with me because the carbon cycle is so important. And the bacillus is a link in the chain in the cycle. So if you think of, a chain that keeps the carbon cycle going, the bacillus, the bacteria, is a link in that chain. So it helps to break down organic compounds. So we talk about organic matter that breaks down. We talked a lot about organic matter today in the soil. Well, how does it actually break down and become nutrients for the plant? We know in organic chemistry, it's all carbon chains and there's nutrients and everything attached to it. And depending how they're put together, they're going to make either plant tissue or sugar or CO2 or water. And so the bacteria kind of help rearrange these organic compounds to build something else. The best analogy I heard for that, somebody once explained it to me like a child playing Legos. You ever play with the little blocks? So, you build a house with your little bricks, and then somebody else comes along, takes all the bricks apart, and then builds a horse with it or something. So the same idea is you have your carbon chains, and that's all the blocks coming together. Well, there needs to be somebody that takes them apart and somebody else that rebuilds them for something else. So that's kind of how the carbon works, where the plant takes the carbon, uses it to build tissue and other things and sugars. So they help convert plant residue into usable nutrients. So once again, the breakdown. So yeah, the bugs and things will come and eat away at it. The fungi will eat away at it, and then the bacteria comes in and kind of cleans up further on. So they support the formation of soil organic matter. So the humus, the humanes, they'll help for the creation of that. And a really important thing here is they feed off the sugars in the plants. So the plant exudes sugars through the roots. Glenn mentioned that this morning. So the plant produces sugars. And as we know, bacteria feeds off sugar. That's how we get alcohol, right? So the sugar feeds the bacteria around the roots. So we have our little colony forming unit, our little endospore. And I said when they have the right conditions, they come out and they multiply. Well, to multiply, they need the sugars. And the plant provides that. So they work together. So then there's a symbiotic relationship where when the bacteria gets active and it starts breaking apart plant material and starts releasing nutrients, it's puffing out CO2 by existing and working hard. And that's when we talked about the EC meter this morning about, the organisms kind of breathing in their heartbeat. And so they produce CO2 when they do that, and that feeds the carbon that the plant needs, makes it available to the plant. So plant feeds the bacteria sugar, and then with that sugar, the bacteria multiplies starts breaking down nutrients in the soil, make it available for the plant, and provides it back with carbon. And so you have your link. And if that's not clear, I have a picture. So it's kind of a complicated picture. I looked for a lot of pictures online because there's so many different carbon cycles, and I found a lot of like fifth grader carbon cycles you could see as well. And if you look, is there light? Whoa, look at that. Okay. So through the roots here, the root exudates, that's where the sugar comes out, and it'll feed your bacteria. And then your bacteria is going to multiply. I got to read this. So here, oh, organic acids, look at that. So you have your amino acids, your fulvic acids, everything starts working together. And so the plant starts feeding the soil, converting the carbon in the soil, giving it back to the plant. Feed to build plant tissue, and then the plant tissue dies, falls back, and then we have that carbon cycle we're talking about. Every time. So in conclusion, for the bacillus, this is a fairly short presentation. I was told half an hour, and I want to leave enough time for all the questions. So we're cruising through this fairly quickly. The Bacillus valensis is a beneficial soil bacterium. The key role that it plays as a plant growth regulator. So it stimulates, it upregulates auxins. I don't know if you guys are familiar with auxins. Anyone? No? Okay, I got a couple head shakes. So a plant has three major growth hormones. There's auxins, gibberellins, and cytokinins. And each one of those hormones is a signaling molecule that tells the plant what to do. So it tells the plant when it needs to grow roots. It tells the plant when it needs to start growing leaves. It tells the plant when to open or close the stomata. And it does a lot of these things. Two other major hormones in the plant are your ethylene and your abscisic acid. Ethylene is your stress hormone. We talked about the ROS earlier. So when the plant is stressed, it starts producing those ROSs. And that keeps it busy so it doesn't get to produce growth hormones when it's producing those. The other one is abscisic acid, which is the dormancy hormone. So when your seed's sitting in the bag and it's not doing anything, abscisic acid is kind of the hormone that's in charge at that point. And then if you ever heard, I don't know if you guys grow potatoes or anything, you might have heard of GA, gibberellins. That triggers the germination. And so often in seed treatments, you'll see a GA in there, gibberellic acid or gibberellin, that helps trigger germination. As soon as the seed has germinated, auxins kick in. Auxins are responsible for root growth. They're responsible for the root architecture of the plant. And that's a really important part because all the other growth hormones are synthesized in the roots. That's where they're produced. So the more roots you have, the more growth hormones you're going to have to promote the growth of your plant, the healthier it's going to be. So, back in a former life, former employer, they described the roots as the brain of the plant. And so that the more roots you have, the more it can do up top. So, the auxin activator is really cool because it produces more roots, which will then produce more of everything. So we're look at it as priming the pump. And then carbon cycling. So it's important for the sustainable agriculture and the soil health, and everything feeds in together. Any questions so far about the bacillus? Am I moving too quickly? We're good? Okay. So endomat expertise, I threw that pretty freely there. That's not verified by anyone. But the reason I wanted to talk about it is because you guys have heard of our products. You've heard of Fosbac 45 and you've heard of Ami Protect 42. And this is not a sales pitch, but I want to show you how the products you might have heard of or you might have used in your fields fit into everything we've talked about so far. So the Fosbac, it's Bacillus valensis. So FZB45 is the name of the strain. It comes in the form of durable spores. So we talked about the colony forming units. It's a biostimulating bacterial inoculant. And it helps with the mineralization of nutrients, and it helps increase microbial activity in the soil. So you can see it there. The unit's kind of blurry. So that the Fosbac 45, the reason it has that name is because its specialty is to mobilize phosphorus for the plant. That's what this particular strain does, the 45 strain. Does it help to prevent against disease and pathogens as well and protect the plant? Yes, to a certain extent. I've done some trials on onions and carrots, and the feedback I got is I didn't have disease this year. I didn't have as much fusarium. I'm like, awesome. It's actually phosphorus, but there are effects. So remember with the biologicals, it's not like a prescribed thing. Like it's not like you have problem A, you put in option B and it solves the problem. So they're more complex. Question?
Is that the same product Exit Bio has?
No, it's different. The XI Bio, is that what you asked? Yeah, it's a different strain. Yeah. Okay. Ami Protect 42. So it's FZB42 strain. It's a sister strain to the 45. And the reason we have both is because the metabolites they make are slightly different. Remember how I talked about when Greg asked, well, which funguses do they kill? Do they kill the good ones too? So the FZB42 was chosen because the secondary metabolites that it produces are toxic to a lot of funguses out there that we don't want, stuff like Fusarium, Rhizoctonia. It also has been proven to have effectiveness against juvenile nematodes, the bad ones. So down in Brazil, it's actually registered as a nematicide. Ensures stronger plants. Do we still get, the phosphorus mobilization from the AMI Protect 42, from the FZB42 strain? Yeah, we do. Is it as efficient as the Phosbac? Maybe not quite as much. So they each kind of have their own specialty, right? So this is a picture under the microscope of the spores. If you were wondering what they look like, here they are. And so yeah, so they're a little more resistant to UV rays. They have a wider temperature range in which they survive. We talked about pH this morning, about the biology that we're trying to keep between 6 and 7. That's for optimal activity. Doesn't mean if your pH goes below 6 that you're necessarily going to kill them. It just means they start checking out. And so in the spore form, they can go down to a pH of 5 and survive. Now, are they going to be super active at 5? Probably not. You want to get that optimal range. And then it makes it resistant to fungicide, insecticides, and herbicides as well, keeping it in that spore form. So their mode of action. So I talked earlier about where they colonize the root and how they can actually protect against pathogens. So this is a picture. You can see the biofilm around right there. I don't know if you can see that. Right there. People on the other side. Over there. A little biofilm. Yeah. So the bacteria starts colonizing the root hairs and the roots so heavily that it doesn't leave room for the nematodes and the other pathogens to get to the root itself. So it creates a biofilm that protects it, a bit of an armor, in other words. It's a natural occurring soil bacteria. So people ask me, they say, hey, is this synthetic? Where does it come from? It's naturally occurring. We're just supercharging our soil. A lot of us are coming off fields that have been used, chemicals been used on them for years. And we talked about it this morning, like we've killed all the bacteria, we've killed the biology in the soil. So we're coming back, we're supercharging that soil with more bacteria. Root-associated growth promoting rhizobacteria. And then, yeah, toxicologically harmless, no pathogen. Function and habitat. So we've talked about rates with Greg a lot because you look at our label and we recommend much higher rates than what a lot of you guys are using. And you might wonder, well, who's right? What's going on, right? And the reality, once again, biologicals aren't straightforward. It's not put a liter of this and it fixes your problem, right? You got to look at it as a system. It's a whole interaction. So companies often, when they put out a rate, they put out a rate based on, you just use this. That's the trial we did. And then you have your control where you have nothing. But if you're a grower that's using amino acids, fulvic acids, humic acids, you're using, your calcium, you're watching your pH, you're making sure your soils are aerated, you're doing all these things, you're putting a lot of pieces of the puzzle together. So chances are you don't need to put 1 billion of these on. If you just put 1 million of them on in the right conditions, they're going to grow to 1 billion on their own. And so that's what we see here in this photo. So we sell these as a seed treatment. that's where I recommend using them. That's where they're most effective. So they're right on the seed. So the moment that seed germinates, remember I said the gibberellins kick in, the seed will germinate, and then it starts producing roots. The moment it's got roots, it's going to start producing sugars, feeding the bacteria. The bacteria is going to upregulate the auxins. It's going to make more roots. It's going to make more room for the plant to grow, more opportunities to make sugars, build more bacteria. The cycle starts going. The pump is primed, and off it goes. And then by the end of the growth, by the time you got a full-on plant, this is a potato plant here, but you have way more than you started with. So the idea is you set your colony, you have the right conditions, and it'll take off from there. Once again, where do the exudates come from? They come in the root tips, the root hairs. That's where the plant provides the most sugars. That's where it's the most vulnerable. That's where the bacteria is going to colonize, protect the most vulnerable parts of the plant, and the places where it uptakes all the nutrients. So that's where it's going to go. So barrier to suppress pathogens, stimulate plant growth and yield. I really like this picture. This is cool because you can see the kind of long bacteria looking thing, and that's a root hair, if you're wondering what the big thing is. So it's not a shampoo commercial, even that's what it looks like. But it's, yeah, you can see how the bacteria is just sitting there colonizing the root hair, and it's pretty densely populated over that. And that makes it harder for nematodes to come through with disease and spread the disease to the plant. Here's another visual for this. And if you had the presentation last year, you might have seen this in Serge's presentation. And I think you can't see this slide too much. They put a dye in the bacteria, so you can see where most heavily colonizates the root hair. So it's really the tips. It's all the fresh growth. It's all the kind of new growth, and that's where it hangs out. That's where the exudates come. That's where the bacteria thrives. So once again, you can see with the dye, you can see it grow on the plant and then move across and just start colonizing in that dye. Any questions? Yeah.
Is it more important to introduce beneficial bacteria, or is it more important to create the environment for bacteria to reproduce?
So question is it more important to introduce beneficial bacteria or more beneficial to create an environment to favor the bacteria? This is where the rate conversation comes in. I think if you can create an environment that's beneficial, I'd still recommend putting some in, otherwise I'm out of work. But it's also, you don't need to put nearly as much, right? Because they will thrive. Now, if you got an environment that's really harsh where they don't survive, you need to bring in a whole bunch of them. So I like the idea of supercharging the soil a little bit, making sure they take over and they outcompete the other ones, so that the refresher. But do you need to have the really high rate? I don't think so. If you have the right conditions, and I think a lot of people in this room, from hearing the conversations I've heard all day, you're the type of people that will have the right conditions. And so you'll get away with a half and half solution. Nothing straightforward in the biologicals. There's no straight answer to anything. So we have the little drawing here, so you can see that there's competition for space. There's only so much surface area on the root mass. So if the bacteria has taken that over, then they can help with the production of auxins, mobilization of phosphorus through the enzymes and increase yield that way. Induction of resistance, like they help protect against the pathogens because there's just only so much real estate on that root. So beneficial effects, I said I'd talk about those metabolites. So we have an example here. I'm not very good at the science words. Last year I didn't want to present because I didn't know enough about this. I've been spending a year and a half now, reading on this, going to training seminars, having internal training within the companies and trying it out in the field and looking at the results that I'm seeing and talking to growers. And so I feel a little more comfortable with it. I'm still not comfortable with these names. Yeah, I'm not even going to go for them right now. But those are the antifungal metabolites. That's what you need to understand, is that there's secondary metabolites that they make, and that's what's actually toxic and giving us the results. So what does it look like in a petri dish? I know you're all wondering about that, so I have the answer here. And I know the hand was about to come up back there. So you can see you have, I believe that's Fusarium in there. and they drop the bacteria, and the fusarium just hates it, right? The metabolites just keeps the fusarium away, and that's how we protect the plant. So once again, the Petri dish, you can see it here. You have the check above, the one that looks like lungs. That's the untreated. And then below, you have your little bacteria just growing in there, keeps the fusarium away. Once again, a lot of people ask me about resistance issues. Because of the biological process, because it's so complex, we haven't been aware of any resistance issues where you can build resistance over time. Like the bacteria will always eat, or the metabolite will always be toxic to that fusarium. We're talking about upregulating here, so the auxin and enzyme production of the bacteria. So the IAA you see in the first row, that's the auxin, and then you have all these other compounds that are upregulated by the bacteria as well. And once again, it's all interactive with the amino acids, the fulvics, everything we've talked about. And then more Petri dish, because I know somebody wanted to see more of these. Once again, up top, we have the control. So when you just let the rhizoctonia kind of go crazy. And then below, you have the bacteria that's been introduced. And then you can see how they just, with their metabolites, they keep the rhizoctonia away. So you have to imagine your seed, your roots, where the bacteria is colonizing. Is it going to go and kill all the rhizoctonia in your field? No, it's always going to be there, but it's going to protect that area around the root mass, protect it from the disease. So fantastic. We've got a miracle product. It's organic. It's great. Andrew loves it. And but there are limitations. And this is where when Glenn was talking this morning, and it really resonated with me, and I really like what he said, and I just kind of want to go through it. So biology lives, like it's alive. So it needs certain conditions to live. And so, sorry for the small writing, but there's a lot of good stuff to talk about. So soil health. So to your point over there, microbiology is dependent on soil conditions. It will help the soil, but needs the right conditions to survive. So yes, your biology will help your soil be healthier, but it's not going to fix it, right? Like you guys need to do that independently. So compaction can be unfavorable to activity. The EC monitor can give you an idea of the activity you have in your soil. So, if you're getting your readings are super low, chances are in your compaction, you're at 300 psi, chances are your bacterial activity is not going to be great. So then you can put a liter of our stuff and might still struggle, even if you put a liter of it, right? Maybe you need to put 2 liters. Maybe no matter how much you put on, it's not going to help because there's just no air in that soil that's not going to get to it. Soil pH. Microbiology is alive and has a preference, 6 to 7. If your pH is lower, it might just go back into a dormant stage into its endosperm and just wait for the pH to be right before it comes back out. Temperature. If it's below freezing, it goes back into the spore form and it just waits for temperatures to warm up. So these are things to keep in mind where, and you kind of went ahead of me with your question, which was great. We need to make sure our soil is in order and then add in the biology, and then that will feed the cycle. And so, the bacteria feed off the sugar. So the more sugar your plant produces, the more it'll feed the bacteria, and the more colony will proliferate. And then the more carbon it's going to go to feed your plant to make more sugar. Once again, the cycle. So to come back to it, Bacillus, they're a piece of the puzzle. Are they a solution to a problem? No. They're part of the solution. They're a piece in the puzzle. As like we talked with Bill, is the fulvic acid going to solve all your problems? No. It's a piece of the puzzle. The amino acids, they really help out, but if you just put those on alone, it's not going to solve your issues. If you have a pH issue in your soil, you got to fix that first. And so they're the link, they're a really important link between soil biology, not soil biology, and the plants. So they keep that cycle going. If you take out the bacteria, all of a sudden you break that carbon cycle, and then that leads to a whole bunch of issues. But if you can have a healthy soil and you throw the right biology in it, then it's going to sustain itself. So here's my little conclusion. This is a quote from me. So Microbes help break down organic chemistry to release carbon and make nutrients available to the plant. If you want more carbon, which we wanted this morning, microbes will help. They're not the only answer, but they're going to help for sure. If you want more microbes, because you just heard my presentation, you're like, microbes are awesome, you'll need healthy soils. So go back to Glenn's presentation from this morning. More carbon will help the plants make more sugar, which will feed the microbes, and the cycle will continue. So that the moral of the story here is there's no single answer, especially when you're talking about biology. So, you go spend some time in the forest, you look around, everything's growing. Everything's just thriving in there. And there's no intervention from us. It's because the complexity has been maintained and it's just thriving in its diversity. And actually visiting Andrew's farm, that greenhouse is really cool. If you ever get a chance to go with the live soil, there's stuff everywhere going, and that diversity is just mobilizing nutrients, favoring all the different microbes and everything. And he's probably got the best tasting tomatoes I've ever had. And that's a result of all those complex systems working together, right? So yeah, if you walk away from today, understand that the bacteria is a piece of the puzzle. It makes the bigger picture. It's not a sufficient condition, but it's a necessary one in there. So if you have any questions, that's the end of it. I'm at half an hour. I was told to do half an hour. I actually went 2 minutes over. So everyone's still awake? Yeah.
I was told I can't grow corn after canola because there's some bacteria that's depleted that takes away the phosphorus availability.
Who told you that? You can grow canola after corn?
Well, they said don't grow canola after corn. They said don't grow canola after corn because it depletes the phosphorus. Corn after canola, yes.
Interesting.
They said don't do it.
You don't usually see them in the same areas. Corn and canola.
They are.
They are. Where are you? Manitoba?
Ontario, you should know that.
Ontario? Yeah, true. Yeah,
You're from Guelph.
that's right. I'd have to look into that one. I'm not familiar with that one
I
particularly.
wondered if that 45 you were talking about, you said it made phosphorus more available.
That's right. So to the point earlier where we talked is, there's a lot of biology kind of competing in there. So if we can come in, set the FZ B45 to establish itself, then it sounds like the bacteria like there's a lack of bacteria, you're saying, after canola? Takes it away for the corn?
Ties up the phosphorus.
It ties it up. So this one would free it up. So you just need to, maybe you'll have to put a little more on to outcompete the other one and colonize your root mass. But once the FZB45 gets on the root, it'll start mobilizing phosphorus for you. So it just needs to outcompete the other one. Have you noticed phosphorus deficiencies when you do that rotation?
They say don't try it.
You didn't try it?
It won't work. So I didn't.
Well, we'll talk. We'll talk. Any other questions?
What was the Amy Protect you said that was protecting against fusarium?
Correct.
Now, is that just on the root or is it the whole plant?
It would be around the roots.
Okay, so have you guys done any testing on whether a foliar applied bacterium would do anything for like fusarium head blight in wheat or anything like that?
Yes. So we worked with it. Now, because of the spore form, once they need the sugar to live, right? So they're more of a soil-based bacteria. So out of the soil, their survival rate's much lower. So it's not great on the leaf. Now I did talk to our manufacturer, and they're working on a foliar formulation where they're either working with a different strain, but it would be a foliar fungicide that is bacteria-based that would survive on the leaf without being a soil-borne. So this, if you take our bottle now that you have in the shed and you put on foliar, probably not going to work very well. Yeah, not setting it up for success.
The Thunderback, what's Thunderback for? Where does it fit into this?
So we talked about temperature range. So that the temperature range of activity with the Fosback and the Amy Protect is around 12 degrees Celsius. That's when the bacteria really starts saying, okay, it's warm enough for me. I'm going to get out the house here, move out of my little pod and actually get activated. Tundraback C5, it's another strain that we have, and that one gets activated at 8 degrees Celsius. So you gain about 4 degrees, which sometimes can mean a few days, a week, depending on your soil temperature and where you live in Canada. So it was developed in Germany, but they really had Canada in mind when they produced it, because they said, well, you guys got cold soils. You're trying to seed in something that's too cold all the time. This is for you. We'll call it Tundraback. So it's got a catchy name, but you gain 4 degrees. I haven't received any major complaints about the Fosback not working in colder soils, but theoretically, according to our trials and according to lab work, the Tundraback will outperform Fosback from 4 to 12 degrees. So you gain that extra bit of time. It's still good at mobilizing phosphorus. It still outcompetes a lot of pathogens. You get a lot of the same. It's just the activation, because it's a slightly different strain, works a little colder. Has anyone here tried Tundraback? Anyone tried compared to Fosback? Do you notice a difference? Not a difference? Yeah. So my guess would be it'd be hard to notice unless you really had that extended period where you had that cool, and then you get an extra week of like activity, and you get those auxins stimulated a week earlier, then you'll get more roots and you'll get stronger emergence earlier on. But you need the right conditions for that to happen. If your soils heat up really quick and it's only 2 days, you might not notice a difference. Anybody else? Sounds good. You guys are tired. Thank you.